Good morning children. Today we are starting next unit, chapter 11, Biotechnology Principle and Processes. Biotechnology deals with the technique which uses living organism and enzymes obtained from the living organism for producing products and making processes useful for humans. That means all processes in which living organisms are used for human welfare can be included in biotechnology. So, all microbes mediated processes like making of curd, bread and wine can also be included in biotechnology. But in restricted sense, biotechnology actually includes the genetically modified organism, and these organisms are used at larger scale for the production of produced or products which are useful for humans. In other sense, biotechnology also includes in vitro fertilization leading to test tube baby, DNA vaccines and modification of genes, removing of undesired genes that also can be included in biotechnology. Now the definition of biotechnology is being given by European Federation of Biotechnology, and it includes both traditional and molecular views. In this, the exact definition of biotechnology is that it is a integration of natural science, organism, cells and molecular analogues for products and services. Services means which are useful for human beings. So now, after discussing the introductory part, we are to now discuss the principles of biotechnology. On what basis the biotechnology is based on. So there are two main principles of biotechnology is genetic engineering. Genetic engineering, that means manipulation of genes, that means alteration of the structure of the gene, or you can say the removing of the undesired part, or you can say the desired part of the DNA or desired gene can be introduced into the host cell. So all this is possible in the field of biotechnology, which we are to discuss in this chapter. Now the second principle of biotechnology is that we are to, or in the laboratory, those conditions are to be provided in which the desired microorganism can be cultured without contamination, or you can say without contamination of undesired organism. This can be explained this way, that in laboratory condition even the sterile conditions are maintained, but even then also some other microorganisms are also present in air, or you can say in the environment. So once we are culturing one microorganism that is considered as desired organism, so that only is to be cultured and it should not be contaminated with any other microorganism. These type of condition are to be provided. So these are the two main principles of biotechnology. Further in this, we are to understand that in asexual reproduction actually no variation arise, or we can say the daughter organism which are formed are the clone to the parent organism. But in sexual reproduction there is no question that why variation arise. Variations are very much certain in sexual reproduction, because in sexual reproduction there is a fusion of male and female gamete, so that there is the zygote which formed is the mixing of the mixing of the genes of both the paternal and maternal organism is there. So definitely the variations will arise into the daughter organism which is being produced by sexual reproduction. So this is just a recalling of the sexual and asexual reproduction, because in that also the variations arises. And in biotechnology also we are trying to making the new organism with some new characteristics. Now one more technique is there, hybridization. We are to actually compare with these processes with the biotechnology. So we are first taking up all those which we already knows. So hybridization is actually the mixing of the genes. Suppose the pollen grain of one of the flower is dusted over the stigma of the other flower. So that way we have mixed all the genes of both the plants, so that in the zygote all the genes which are present are the mixing of both the plant. So which gene is appear in the daughter organism, it all depends upon chance occurrence. Suppose the desired gene will not appear, it all depends upon repeated experimentation, and once we will perform repeated experiment, then only it is possible that desired gene will appear in the daughter organism, and sometime it may not appear. But in biotechnology, actually what is there, that the genome of the organism is studied, and from there the desired gene can cut with the help of the enzymes, that those are called as molecular scissors. With the help of restriction enzyme, which are called as molecular scissor, the desired gene can cut from the desired plant or desired organism, and those can be introduced into the host plant by also extracting the DNA of the host plant, and it also can be cut with the restriction enzyme, and desired gene can be introduced into the host organism, and it can be linked there with the help of another enzyme that is called as DNA ligase. That means biotechnology is such an advanced process in which the genes can be cut and can be pasted and can be introduced into the another organism, and that is only the desired gene. It's not required to mix all the genes in biotechnology, so that it is a advanced technique over sexual reproduction and over hybridization. That's why this comparison is given. After that, it is given that this technique is actually developed by Stanley Cohen. How have they developed? They have taken, so first of all, the Salmonella typhimurium. This bacteria is actually resistant to the antibiotic. So that they have studied the genome of this bacteria, and then they have taken the gene which is present in the Salmonella typhimurium, which provides the antibiotic resistant characteristics to this bacteria. Now this gene is been cut from this bacteria, and one more thing is there, that is called as plasmid. Plasmid is actually called as extra chromosomal DNA, which is present in the bacteria. So this plasmid can act as a vector. Okay, in biotechnology there is a rule, there is a use of vectors is there. Like you can compare it with the mosquito, which carries the plasmodium from one person to the other person, and the mosquito act as a vector. So similarly here the gene can be transferred through the vector, which is called as plasmid. That means that gene will not directly introduce into the host cell. First it will be ligated with the help of DNA ligase into the vector. First the vector will be cut by the help of restriction endonuclease enzyme, and that gene will also be cut from the desired organism, and that gene will now be introduced first into the vector and will be ligated with the DNA ligase enzyme, and after that this recombinant DNA will be ready, or it is this plasmid in which the desired gene is introduced, now called as recombinant DNA, and this technology is called as recombinant DNA technology. And now this recombinant DNA will be introduced into the host cell, and Stanley Cohen has used the Escherichia coli, which was not antibiotic resistant, but once the antibiotic resistant gene of the Salmonella typhimurium is introduced through the vector into the E. coli, so E. coli has also become antibiotic resistant. So this has shown that through the biotechnology, one can introduce the desired gene into the host cell, and it is to be linked with the host cell. It is to be linked with the chromosome of the host cell, and it is to be linked with the chromosome of the host cell where at the point where there is a origin of replication is there. In the host DNA, there is a point where origin of replication is there. There the foreign DNA or aligned DNA is ligated, so that now the foreign DNA or aligned DNA will multiply along with the host DNA. That way one can prepare the multiple copies of the host DNA, and this process is called as gene cloning, or you can say the gene library can also be prepared. So these all are the actually the principle of biotechnology are there, which I am going to discuss you in the book also in the next video. Thank you.